Hello and welcome to the inaugural Karyotalk webinar, The Nuclear Pore in XPO1, Guardian of the Genome. I'm Tina Beamon, Vice President and Chief Compliance Officer with Karyopharm Therapeutics. We are delighted to have you as a guest for today's event. Today's disease state program will explore the role of exportin-1 protein, also called XPO1 or CRM1, in the nuclear pore complex and in nuclear transport. The nuclear pore complex is made up of a multifaceted network of biological actors, including importins that bring proteins from the cytoplasm into the nucleus, and exportins that bring proteins in the other direction, from the nucleus out into the cytoplasm. Over the next hour, we will learn about the nuclear pore complex generally and how XPO1 protein, one of the key exportins, behaves within this framework, including its impact on a variety of different diseases like cancer, COVID, traumatic brain injury, and Duchenne Muscular Dystrophy. The presentation will begin with an introduction from Karyopharm's President and Chief Scientific Officer, Dr. Sharon Shacham. We will then hear from panelists, Professor Michael Rout, Professor Cesario Borlongan, Dr. Thomas Walsh, Professor Matthew Alexander, and Dr. Clifton Moe. Please turn your attention to the menu at the bottom of the screen. We will release polls throughout this program. Please vote on the answer that best suits you. Today's polling will help us understand what parts of the program are most important to you. So with that, let's get started. Please welcome Karyopharm's President and Chief Scientific Officer, Dr. Sharon Shacham.
Thank you very much, Tina. And I should mention Tina is our head of compliance, so this way we are making sure that we are all compliant here. And this is a very exciting webinar. It's our first webinar in the series of the Karyotalk. And I would like to take this opportunity to again thank all the panelists for joining us in this very first webinar, and to the KaryoNext team that worked and enabled us to have this webinar. We are going to have a very special journey today. So one of the most interesting pathways in biology. And together with my colleagues, we will show how one protein in the cell can play a very important role in many diseases, as Tina mentioned, from cancer to brain trauma, to DMD, and to viral infections. The protein that we are talking about is called XPO1, and it's like a train. And although we can't really imagine our life without an efficient train system, and I'm not talking about Amtrak here, but a real train system, sometimes the XPO1 train is being kidnapped by the disease and it gets out of control. And when that happens, it threatens to damage the cell. And today we will discuss what are the situations that this train is getting out of control. And we can see the little monster here of the disease. And one approach that we developed here at Karyopharm to bring the train back into control and continue the path of a normal cell. I know some of you might still not understand what am I talking about looking at this cartoon, but that's the idea. So you will stay with us until the end of this webinar. And at the end, there's going to be a quiz, so I'm sure you'll all know it by the end. So if we can move to the next slide. So Karyopharm was founded in 2008 by the peculiar Dr. Shacham. And this was about, by now, 12 winters ago. And we are just heading to our 13th winter here in Newton, Massachusetts. And the idea was really started by a paper that Professor Rout, that is going to speak after me, published, which showed the first high resolution structure. It was a model of the nuclear pore complex. And with that, we thought that if the thought came to us that if we can play with what's going in and out of the nucleus, we can really impact one of the pillars of the protein, which is the communication between the nucleus and the cytoplasm. The company had, we started to develop small molecule completely from scratch. To be honest, some of this work was done on my computer, and the compounds were stored in our home kitchen in the refrigerator, and we found a few hits. We developed them, and in 2010, we were able to demonstrate for the first time anti-cancer activity in vitro and in vivo. And after that, we started clinical trial in 2012, so just about 8 years ago. And this was in patients with blood cancer and with solid tumors. Since then, we have treated more than 3,000 patients, including about 150 kids with selinexor as single agent and in combination. And recently, we also treated about 150 patients with severe COVID-19. So this was in the last 8 years. We got our first approval for selinexor just about a year ago in July 3rd of 2019, and that was for patients with pentarefractory myeloma, so very refractory multiple myeloma. And about a year after that, we got our second approval, this time in patients with relapsed refractory DLBCL. And as you can see on the slide, and we'll might be able to mention it a little bit after Dr. Moore will have his section talking about cancer, is that this mechanism of action might be applicable to many cancers as well as other diseases, as we are going to talk today. So if we'll move to the next slide. So in this slide, we are seeing on the left data from brain tumor, from glioblastoma, which is the most aggressive form of brain tumor. Now, I want to remind you that cancer in general is associated with unregulated cell growth. And on the right, we are seeing data from patients with multiple sclerosis or from a model of traumatic brain injury that is done in rats, and these two models are associated with cell death. But what is combined to these two situations, or two different or three different diseases, is that in all of them, we see an increase in XPO1 or CREM1, that's the other name of this protein, that is coming in the situation of the disease compared to normal cells. We see it here in glioblastoma, and we see it here in patients with multiple sclerosis compared to patients without. And this in post-mortem analysis. And we see it also in the first few days after traumatic brain injury. In all of these cases, either associated with unregulated cell growth or with cell death, we see an increase in XPO1, which is very interesting. On top of that, we can also know, and we have seen it in other cancers as well, is that the more XPO1 you have, or more CREM1 you have, the survival is lower. So higher CREM1 is associated with increased mortality. Why is this? How can one protein be associated in both cases? Let's go to the next slide and try to understand. First, what is XPO1 and what is it doing? In this cartoon, we see on the right the nucleus of the cell, and on the left, the cytoplasm. The transport of protein through the nucleus to the cytoplasm and vice versa is highly regulated, as we can expect that. And for protein to go in and out, all protein above 40 kilodalton, they have to go through the nuclear pore complex, one of the most complicated structures in the cell. And Professor Rout after me will talk about this in details. However, this transport across the nuclear pore complex can only be done through carriers or trains. And XPO1 is the main train that takes protein from the nucleus into the cytoplasm. We see it in this gray bagel, and the cargo is seen as the green part. It releases it in the cytoplasm, and it goes back in to pick up another cargo and send it out to the cytoplasm. And it can do it multiple times. The more XPO1 we have, the more nuclear export we see. So how does it look? Can we go to the next slide? What is happening, or maybe I'll take one step back. There are about 200 cargoes that XPO1, mammalian cargoes, that XPO1 can take from the nucleus into the cytoplasm. And there are other cargoes like viral protein that Professor Walsh will talk about it. But let's focus for a second just on the mammalian and the cellular cargoes. Amongst these 200 protein, it includes all the tumor suppressor protein, which we also call them the guardian of the genome, because that's exactly what they do. They sit in the nucleus, they monitor the DNA for DNA damage, which is the hallmark of cancer. And once they identify DNA damage, they say, okay, this audit essentially failed. They call the cell, they induce a cell cycle arrest and induce apoptosis of the cancer cell. When XPO1 is overexpressed, I don't know if there's a chance to get the cartoon again. It takes the tumor suppressor protein, and what we can see in cancer cells that the tumor suppressor protein are actually outside of the nucleus and not where they are supposed to be. So the train takes them out of the nucleus and release the guardian of the genome or the tumor suppressor protein into the cytoplasm where they cannot do their work, and that's how the cancer cell can proliferate. Our thought was simple. If we will block this train, we will block the excessive export, nuclear export of this tumor suppressor protein, will restore their localization into the nucleus, and they will go back to do their job to identify DNA damage and send cancer cell into cell cycle arrest. And that's exactly what the SINE compounds are doing. They bind to XPO1 in the cargo binding domain, and it inhibits nuclear export. So how did we do that? If we go to the next slide, what we see is the X-ray structure of XPO1. The X-ray structure of XPO1 was published by Professor Yumin Chuk from the University of Texas in Dallas in 2009, really when I started the company. And this structure really enabled us to identify small molecules that will bind to XPO1 and prevent cargo binding. So it's a very complicated protein. We can see it here. And in the right side, in the upper right side, we can see the cargo binding domain. So there is a helical structure or a cavity that allows helical structure to bind, and many of the natural cellular or other natural cargoes of XPO1 have this helical structure. Interestingly, in this upper here, what we see at the bottom, there is a cysteine residue that is conserved all the way from yeast to eukaryotic cells into our cells as well. And this cysteine is not part of the cargo binding domain. It doesn't participate, but it's still very conserved, and there are all sorts of assumption why is it there. But what we did is we used the cysteine and identify a small molecule that will bind to the cysteine, and we can see here 2 examples of SINE compounds. And by that, they block the cargo binding domain and prevent the regular cargoes of this strain to bind to XPO1. And then they left in the nucleus and do their function. So how does it look in the cellular level? So this work was done by our team with one of the early compounds. We see here osteosarcoma cells, and we see about 4 hours of treatment with a SINE compound that we squeezed to about 10 seconds of a movie. We'll see when the movie starts again is that this protein that we look, which is one of the cargo of XPO1, is actually everywhere except the nucleus. So this is a tumor suppressor protein that's supposed to be in the nucleus, and in cancer cell it's being exported to the cytoplasm and cannot function in the nucleus where it's supposed to be. Once we treat these cells with our XPO1 inhibitors, we can see marked nuclear localization by this intense color, like we see here, of FOXO, the protein that we will follow in the nucleus. And now this protein can start functioning and induce apoptosis in cancer cells and cell death. Now, the same protein in other diseases, like in multiple sclerosis, for example, is essential to prevent cell death. And again, in the situation of the disease, it's excluded from the nucleus. So restoring this same protein function in the nucleus in other diseases will prevent the progression of the disease. So this is a brief introduction. So if we go back now to our cartoon about the train, I'm going to give you a few more hints, and let's summarize it when we look. So we have the XPO1 train expressed in all our cells, essential for the function of normal cells. However, in a situation of diseases like cancer, multiple sclerosis, TBI, and it's also essential for viral propagation, we see more trains, and we see the train being essentially kidnapped by the disease. And that's leave the tumor suppressor protein just going to be kidnapped, carried by this train and move outside where they're supposed to be. So this is our superheroes here, getting out from where they're supposed to be into the nucleus and pushed with the train outside of the nucleus. And the little figure here is the SINE compounds that prevent that and stop the excessive trafficking induced by the XPO1 train. So I hope that helps. And now maybe we'll take one step backwards, and we'll let Professor Rout explain about the entire bigger system of the nuclear pore complex. Tina?
Thank you so much, Dr. Shacham. We are pleased to now introduce Professor Michael Rout of the Rockefeller University, who will present on the nuclear pore complex.
Hey, thank you very much. I'll just see if I can get this to work. There we go. Hopefully you can all see that slide. Yes?
Yes.
Great, thank you. So thank you very much for having me here. So I'm going to take a step back, actually quite a few steps back, a sort of 30,000-foot view and a very brief summary of the work of many groups over many years from all over the world, trying to piece together what the nuclear pore complex looks like and how it functions in transport of materials into and out of the nucleus. So we're starting, as you can see, with the nucleus district. It turns out it's not only in the cell, it's a place in Montana, as you can see here. But the nucleus is where all the genetic material of the cell is, and bounded by a double membrane made of an outer membrane and an inner membrane. Where those membranes meet in circular grommets is called the nuclear pore, and there are many of these nuclear pores all over the nucleus, but they're not just open, they're filled with a proteinaceous complex called the nuclear pore complex right here. And as you've heard, the nuclear pore complex's job is to be the gatekeeper of the nucleus, to bounce out anything that is not supposed to be in there, to keep in things that are supposed to be in there, and to make sure that the things that need to go in get transported in, the things that need to go out are transported out. Small molecules like sugars and ions actually can diffuse passively into and out of the nuclear pore complex, but macromolecules, and in particular proteins and RNAs, have to be actively transported in what is known to be a highly selective and highly regulated set of processes. So the many different kinds of macromolecules are imported and exported through nuclear pore complexes. This is an electron micrograph section of a typical eukaryotic cell. The nuclear pore complexes are these very, very small things here, but there's thousands of them, like 2,000 or more on an average mammalian cell. For example, ribosomal proteins must be made in the cytoplasm, imported into the nucleus, where in the nucleolus they get packaged into pre-ribosomal subunits, and those pre-ribosomal subunits have to be exported again out of the nuclear pore complex, where they get assembled finally into the full ribosome and so on. And of course, messenger RNAs are made in the nucleus. They're then packaged into RNPs and exported through the nuclear pore out into the cytoplasm, where they then get translated, and the proteins that carried them get recycled back to carry more mRNAs. And so, in fact, the central dogma of life, DNA makes RNA makes protein, is in fact interrupted by the nuclear pore at the RNA makes protein stage. The RNA has to get out into the cytoplasm. But what we're particularly interested in today is the import and export of proteins. In order to for a protein to get into the nucleus, it has to have a little signal on it. So the cargoes must have a little signal called an NLS, a nuclear localization sequence. This gets recognized by any one of a member of a family called Karyopharms, or importins or exportins. Because they were independently discovered by several groups, they still carry these different names. An importin Karyopharm binds this NLS on the cargo protein and then will carry it through the nuclear pore complex out into the nucleus. In the nucleus, it meets this protein called RAN-GTP. That is, RAN is a member of the RAS superfamily of GTPases, and it can exist either in a GTP-bound form or a GDP-bound form. It's maintained in the GTP-bound form in the nucleus and the GDP-bound form in the cytoplasm, and that energy differential is what powers transport. You can think of it like a hydroelectric dam with the high energy all in the nucleus and the low on the other side, and the bleed of that RAN energy is what powers the cycle. If you are a protein to export, you have a little signal on your cargo protein called an NES, or nuclear export sequence. And there are a cognate family of Karyopharms that recognize those NESs and export the cargoes out through the nuclear pore. When they get to the other side, the RAN-GTP that's bound to them hydrolyzes to RAN-GDP, and it's that burst of energy, as I say, that then powers transport. And RAN itself has its own little transport factor that takes it back into the nucleus, where it gets regenerated into the RAN-GTP bound form, and the whole cycle then continues. But it's all centered on this huge macromolecular complex called the nuclear pore complex. I'm going to show you a quick overview of some of the major structures of the nuclear pore. They're made of a membrane ring, which is in the lumen of the nuclear envelope. It sort of holds the whole nuclear pore into the curved grommet that you can see here of the membrane, where the outer and inner nuclear membranes of the two membranes of the nuclear envelope meet. That attaches to this very dense inner ring complex, which is flanked on both sides by an outer ring complex. And then attached to those are peripheral machineries on both the cytoplasmic and nuclear plasmic side. In this diagram, the cytoplasm is to the top, the nuclear plasm is to the bottom, and they're used to help assemble and disassemble transport cargoes with their transport factors. Studied throughout the tube in the middle, the tube that all the stuff goes through, in and out, through the nuclear pore, are these anchors for these things called FG repeat nucleoporins. They're called that because they actually have phenylalanine glycine, that's FG repeats, dozens and dozens of them in each protein, and they form this dense, wiggly, dynamic network of highly dynamic, brush-like, dense region going throughout the whole tube of the nuclear pore. As you can imagine, that forms a barrier that most things can't get through, but transport factors, Karyopharms and other transport factors, with their cargoes, the transport factor can bind the phenylalanines on those FG repeats. They have a series of little pockets that combine the phenylalanines, and they do so very, very quickly on and off. And by being able to do that, they can overcome this dense meshwork that normally keeps stuff out and actually carry their materials across the nuclear pore, either into or out of the nucleus. So I'll just give you a quick movie here of just to give you some idea of the assembly. The whole thing here is about 100 nanometers across, the hole in the middle filled with this selective meshwork that will only allow transport factors through, it's about 40 nanometers across. The whole structure is anywhere from 50 megadaltons up to over 109 megadaltons in most human cells. This is a yeast one, but the vertebrate one's even bigger, but the principles are about the same. You can see it's made of these coaxial rings at different levels, and that there are, in fact, 8 spoke units making up these coaxial rings. But what we found by looking at the structure of the nuclear pore is it's very much like reminiscent of a suspension bridge. In that, the membrane ring I just showed you firmly anchors it into its substrate, which is the curved membrane of the nuclear pore. Inside each spoke, and by the way, the view I'm giving you here in these little diagrams is as if you were inside the tube looking out towards towards the spokes, and you're seeing 3 of those spokes just looking out at them. So it's from the view of an actual thing going through the central tube. You can see that these spokes have these rigid columns, just like the supports and trusses of a bridge, but that between these rigid supports are flexible joints, because like a suspension bridge, you need flexing to allow the bridge to breathe and accommodate heavier or lighter traffic. Likewise, all the major units in the bridge are connected by flexible connector cables. And again, they are tense to allow tremendous strength, but also allow flexibility as bigger or smaller things go through the nuclear pore. And of course, all of this builds this high density transport pathway, anchors these FG repeats, points them into the central tube in a very organized fashion to create pathways, highways of binding through the nuclear pore that transport factors can carry their cargoes. So this all builds a very strong and flexible transport system that can maintain this huge transport flux in both directions. So that's sort of a quick overview of the nuclear pore complex. But as I said, just like a suspension bridge, there are many different kinds of traffic going through the nuclear pore complex, and different Karyopharms specialize in carrying different kinds of cargoes, either in or out of the nuclear pore complex. We're still trying to understand what all these different cargoes are, and we haven't got a complete list of who is carrying what. But there are certainly a whole series of import Karyopharms that carry stuff into the nucleus, and they're different kinds of proteins that can be carried. So some, for example, specialize in carrying only ribosomal proteins, some specialize in carrying H and RNP proteins, and so on and so forth. And export, likewise, there are some that specialize in, for example, tRNAs, certain kinds of proteins. And of course, the one we're interested in today is a bit of a universal carrier, CREM1 or XPO1, which recognizes any cargo that carries these NES, typical NES sequences. And that's the quick overview, so we'll leave it there. Thank you very much.
Thank you so much, Professor Rout. That was a fantastic presentation. We're getting some questions in the chat feature, and we're going to just ask you to hold your questions until the very end, and should we have time, they will be addressed at the end of the program. So with that, I'm going to invite up our next speaker, Professor Cesario Borlongan of the University of South Florida, who will present on the role of XPO1 in traumatic brain injury.
Thank you, Tina. So I'm Cesario Borlongan. I'm from the University of South Florida, Morsani College of Medicine, and I'm a director of Center of Excellence for Brain Repair and Aging. My interest in XPO1, I would focus more on the traumatic brain injury. And I'm trying to move my slide. Okay, thank you. So just for disclosure of conflict of interest, I received funding from Karyopharm some years ago for this specific project on traumatic brain injury. Next slide, please. Many of the data that I will present today have been published some years ago on this journal. And again, trying to segue from Dr. Shacham and then Dr. Rout's previous presentations, I would focus more on, rather than trying to sequester the cancer signals in the nuclear pore, I am very much interested in trying to sequester the inflammatory signals associated with neurological disorders such as traumatic brain injury. As you know, even though traumatic brain injury is considered as an abrupt brain insult, there is a secondary cell death plagued with inflammation. So that's my bread and butter, is to see how we can sequester that secondary cell death associated with traumatic brain injury. Next slide, please. So the impetus for this study, as alluded by Dr. Shacham earlier, is that in cancer cells such as the THP1 or the HeLa cells, you could see that under controlled conditions, many of these cargo proteins are in the cytoplasm or in the membrane areas following treatment with the SINE or the KPT350. You can appreciate that many of these proteins now get sequestered into the nucleus. So just like what Dr. Rout presented earlier, is targeting that nuclear pore, you are able to control and regulate the export of these proteins, in particular the inflammatory proteins. Next slide, please. So in the initial communication with Karyopharm, I was presented with this data. So this is an in-house company data that when you treat prior to experimental traumatic brain injury in mice or in rats, you could see that the KPT significantly reduced the brain injury at the gross morphology. And so being a translational researcher, I would rather initiate the treatment post-injury rather than pre-injury. So that's my contribution to this project. On the subsequent sets of slides, rather than initiating the XPO1 inhibitor or SINE, we initiated the treatment post-injury. And again, you can recognize here, following the traumatic brain injury, which is a cortical controlled impact injury, there's a significant damage into the cortical region of these rats. Following transfusion or infusion of the low dose or high dose of the XPO1 or inhibitor or KPT350, there's a significant reduction in the cortical damage. And this is not just in the impact injury, but if you count the number of cells lost in the peri-impact injury, so these are like the area surrounding the core injury, there's also a significant reduction of cell loss. So this is quite a significant finding. Again, I want to reiterate the treatment of the XPO1 inhibitor or KPT350 was initiated post-injury, starting at 2 hours, and then subsequently the animals receive over the next 4 days, once a day of this treatment. So this has some kind of clinical implication. As you know, we cannot predict when TBI will occur. So for us to be able to initiate the treatment after the injury, I think this will have a significant clinical application. Next slide, please. Now, not only did we find that the reduction in brain injury is significant, but there's also a corresponding recovery in behavior. So these are just two of the behaviors that we conducted. As you know, we targeted one hemisphere with the CCI, with the TBI injury. So there's a significant hemi-neglect, as you can see also in patients. But these are in animals. When you subject the animals to a rotarod test or the tail hang test, there's an asymmetry in their body movements. And again, we can appreciate with the treatment with the KPT350, there's a significant improvement in their asymmetrical behaviors, indicating there's a significant behavioral recovery from traumatic brain injury. Next slide, please. So in terms of the histology, not only did we see a significant reduction in the gross morphology, but also if you look at the blood-brain barrier markers, in this case, Evans blue extravasation, there's a significant reduction in the entry of this compound into the brain. indicating that the blood-brain barrier integrity remains intact in animals that receive the KPT-350. And other markers of the blood-brain barrier, such as the beta-catenin, also show that in animals that receive this compound, the XPO1 inhibitor, there's a significant preservation of that tight junction proteins, such as the beta-catenin, that would indicate also that the blood-brain barrier is preserved in animals that receive the XPO1 inhibitor. Next slide, please. As I mentioned in my introduction, my interest is trying to look at the inflammatory signals which is accompanying the secondary cell death of TBI. And lo and behold, what we found, the markers of inflammation here demonstrated by TNF-alpha is also significantly reduced following the treatment with KPT-350. So this indicates to us that there's a sequestration of these inflammatory signals within the nuclear pore, and we are able to sequester also the cell death associated with inflammatory response in TBI. Next slide, please. So just to summarize what I presented in these animal models of TBI, there's a significant reduction in the cell death that we're seeing, the secondary cell death. It's associated with anti-inflammatory signals as well as the neuroprotective signals within the nuclear pore. This type of sequestration of cell death is leading to functional recovery in TBI animals. And this is also associated with blood-brain barrier integrity. That type of mechanistic of actions are correlated with reduction in inflammatory signals and blood-brain barrier integrity. Next slide. So in the current times, we are also interested in trying to extend this type of nuclear sequestration in COVID-19. And the next speaker, Dr. Walsh, I don't want to steal the thunder from Dr. Walsh, but at least from our end, we saw that in some of our patients with COVID-19, not only are we seeing inflammation in the lung, but also in the brain, especially in patients suffering from stroke after COVID-19. And so the significant increase in the patients with COVID-19 in stroke incidence could be associated also with the inflammatory response. Our hypothesis is if we could hinder that inflammatory response associated with COVID-19, there is a high likelihood that we may be able also to ameliorate that secondary cell death of stroke that's associated with COVID-19. So thank you so much. I'll give it back to Tina. Thank you.
Thank you, Professor Borlongan. And as you previewed for us, our next speaker will be Dr. Tom Walsh of Weill Cancer Medicine, who is an expert in infectious disease. Dr. Walsh will be presenting on the role of XPO1 in COVID-19.
Good afternoon, and thank you all for the opportunity to talk and to discuss this important disease, especially as it relates to the potential therapeutic intervention from selinexor and other SINE inhibitors. I would preface just by placing in context the COVID-19. Our mission here at Weill Cornell Medicine is that of developing new therapeutic interventions for life-threatening infections, and that in our immunocompromised patient population. With the advent of COVID-19, Karyopharm had contacted Dr. Ruben Novetsky, who has done landmark work in the area of multiple myeloma with selinexor. Dr. Novitsky then contacted Dr. Small and me, where we also are boarded not just in infectious disease, but also in oncology, and hence a very strong translational partnership developed with the three of us, then working very closely with Karyopharm and helping to develop protocol initiatives and helping to further understand the implementation of selinexor as a potential anti-COVID-19 compound. And so with that, I'd like to provide you then with that context the background for COVID-19. Where we stand literally as of yesterday, the epidemiology globally shows that the severe acute respiratory virus syndrome, which is the virus itself, SARS-CoV-2, is causing a global pandemic of coronavirus 2019, the disease being COVID-19, resulting in more than 24 million infections and sadly, more and tragically, more than 800,000 deaths worldwide. Next slide, please. From a practical clinical standpoint, we see a very distinctive pattern upon initial presentation, manifested initially by cough, dyspnea, fever, and hypoxemia. COVID-19 may progress to a lethal, potentially lethal cytokine storm, deadly respiratory disease, and multi-system failure that is characterized by a striking pro-inflammatory response. Early in the disease, we recognize that there are two phases, one of which is the early manifestations of what seem to be consistent with a viral infection, but soon evolving, particularly in more compromised patients. As we know, the elderly, very elderly, patients with diabetes, other comorbidities, into a striking inflammatory process that cause multi-organ dysfunction. So recognizing those two phases, as we'll discuss, selinexor, or the concept of XPO1 inhibition, may offer a powerful therapeutic target. Next slide. Thank you. So conceptually, if we want to then target nuclear export as a novel mechanism to treat viral infections, considerable work had already been done, as with a number of the SINE molecules, particularly verdinexor, selinexor, and other agents as antiviral therapeutics in the last several years. Most investigational agents, if we come back to COVID-19, however, inhibit either the viral cytopathic effect or the pro-inflammatory response. And there have been many protocols that have been brought to bear, usually again targeting one element or the other element. Selective inhibitors of mammalian nuclear export, i.e. SINE, such as selinexor and verdinexor, inhibit both the viral proliferation and block, especially the NF-kappa B mediated pro-inflammatory cytokine release. In understanding the viral cytopathic effect in this early work that was done with Venezuelan equine encephalitis virus, one can see, looking at DAPI stains and capsid stains, and then cytoplasmic stains, one can then see the accumulation with the exportin inhibitors, KPT-185, 335, and 350, using leptomycin as a control and DMSO as controls. You can see the accumulation of viral product within the nucleus. But conceptually, and if you look at the right panel, look into the nucleus in the aqueous blue and the little circle of molecules below. To the left, you have viral protein cargo. And look at the spectrum, the striking spectrum of viruses, from HIV, Rev protein, influenza, the nucleocapsid protein, from JC virus, acnoprotein, human cytomegalovirus, PP66, HPV, as well as kappa C sarcoma, their own distinctive proteins. So on the one hand, there is, through the XPO1 inhibition, the potential for blocking these particular viral proteins and shutting down the viral cycle. At the same time, to the right, there are multiple molecules, host molecules that are exported, but very critically, those involved in the NF-kappa B driven inflammatory cascade. Next slide. So if we want to then focus for a moment just on the viral replication, again, over the last several years, a number of investigators have done outstanding work in understanding the potential role for selective inhibitors of nuclear export. And as we know, these compounds attack the nuclear retention of leucine-rich nuclear export signaling proteins. They block the export of cargoes for a number of proteins, including SARS-CoV-2. and with that disrupt the viral replication process. In the panel to the right, this is a model, for example, for influenza, where we see the nuclear capsid protein, as well as viral ribonucleoproteins being inhibited by the XPO1 inhibition. Next slide. So here, in a series of experiments conducted by Dr. Tripp and his team at University of Georgia, we see in the murine model for influenza, the H1N1 model, the study looking at the role potentially in vivo, not just in vitro, but in vivo, of altering the natural course of viral murine influenza. With that, we see that this follows a pattern similar to what we might anticipate for some of the other viruses, respiratory viruses such as RSV, adenovirus, but with a focus on influenza, one can see that initially, if one targets different points of delay, day 1, 2, 3, and 4, progressively using plaque assay, one can demonstrate in the lungs more than a log drop of virion. This translates very nicely then into improved survival, decreased pulmonary injury, and decreased burden of influenza in vivo. Next. So in correlating that, is there a way that we could then look potentially at SARS-CoV-2 as a potential target? Well, Dr. Zhao and colleagues have done elegant work in this area in several different studies, but in a functional proteomics study, examined the potential for XPO1 as one of the proteins with the highest functional connection to SARS-CoV. And in the little red circle, you can see the central location for XPO1 for multiple viral proteins, suggesting that blockade leads to inhibition of coronal viral assembly proteins. And indeed, earlier work in non-SARS-CoV-2, but SARS-CoV demonstrated that protein B or 6 also led to significant reduction in antiviral activity. Notably also, if you then look in separate experiments, one can see anti-inflammatory transcription factors such as I-kappa B and PPR gamma are also affected. Next slide. So if we then ask the question, well, can we also demonstrate this effect in SARS-CoV-2 viral propagation? Going back to our Vero E6 cells, if they're pre-treated with selinexor or SARS-CoV-2, one can see either with treatment or with prophylaxis or with treatment, significant reduction in the plaque assays with very tight replication and repeated experiments taken out over the course of as long as 96 hours. These in vitro data certainly lay I think a powerful foundation then for going forward experimentally and clinically. Next slide. So if we ask the question then, well, given the potential antiviral effect, what is the potential benefit of the anti-inflammatory effect? Again, trying to exploit both mechanisms of both antiviral and anti-inflammatory effect of selinexor, or potentially even other molecules of inhibition of XPO1. In looking at the pro-inflammatory response in patients suffering from COVID-19, the easiest way to understand this is to simply look at the pattern of inflammatory cytokines across the bottom: IL-2, IL-4, IL-10, interferon gamma, TNF-alpha, and IL-6, all of which show a striking pro-inflammatory response in healthy controls versus those that are COVID-19 with ICU or no ICU. So the last two rows or columns in each graph demonstrate the significant increase in IL-2, especially, and interferon gamma-driven process, and IL-10, and strikingly so for TNF-alpha and IL-6. So given this vast upregulation in the inflammatory cytokine storm, especially associated with intensive care unit. Therein lies the potential for further intervention. Next slide, please. So is there a way that we can then understand that in an infectious process? And the answer is yes. If we look at, again, going back to the H1N1 model, we can see within lungs following 4 days post-infection and then treatment with selinexor in this situation, in comparison, say, to oseltamivir, there's significant reduction of intrapulmonary interferon gamma, TNF-alpha, and IL-6. Next slide. And similarly, if we want to then take a step back and say, well, what is the effect on overall sepsis? Some of our poor patients with COVID-19 that we cared for in the intensive care unit had such striking pro-inflammatory response that an association with the multi-organ system failure, there was almost a process reminiscent of that of sepsis. So if we look back then at the inflammatory LPS-induced sepsis murine model with selinexor at 15 milligram per meter squared, we see significant reductions in the LPS compared to the LPS selinexor treated animals, significant reductions in IL-6, TNF-alpha, as well as recruitment of intraperitoneal macrophages and neutrophils. And next slide, please. I think Tina stepped up. There we go. Good. So with that powerful preclinical background, and as well as the safety profile with a substantially lower dose, lower than that of the antineoplastic dose, with 20 milligram tablet treated on days 1, 3, and 5. We then embarked upon a prospective double-blind randomized control study of phase 2 of hospitalized patients who were greater than or equal to 18 years of age. With that, patients were randomized 1-to-1, either to selinexor on the regimen outlined for the potential not just of 1 week, but if they were doing well for 2 weeks, versus an oral placebo on the same regimen days 1, 3, and 5, and then potentially for up to 2 weeks. Our primary endpoints then being the TTCI, which you see here, which is a time to clinical improvement that would include absence of fever, temp less than 38, and one of the following, either reduced tachypnea, improved oxygen saturation, or hospital discharge. As exploratory secondary endpoints, we also evaluated safety, all-cause mortality, and several other addition markers, both for inflammation and oxygenation. Next slide, please. So in summary, COVID-19 may progress to a devastating cytokine storm, lethal respiratory disease, and multi-system failure. Most investigational agents against COVID-19 either inhibit the viral cytopathic effect or the pro-inflammatory response. Selinexor, a selective inhibitor of mammalian nuclear export, SINE, inhibits both the viral proliferation and blocks NF-kappa B-mediated pro-inflammatory cytokine release. And as an oral agent with extensive preclinical rationale for antiviral and anti-inflammatory activity, SINE compounds clearly warrant further investigation as potential treatments for COVID-19. Thank you so much.
Thank you, Dr. Walsh. I'm sure you're noticing the time. We're scheduled to end this presentation at 2:30. However, we have a lot of great content still to come. So if your schedule permits and you can stay on with us, we invite you to do so. That being said, if you have to go along, we completely understand. This session is being recorded. So as with any good superhero movie, we do not intend to leave you with a cliffhanger. So to the extent that you would like to access that recording, that will be available for you as well. Our fourth speaker is Professor Matthew Alexander with the University of Alabama at Birmingham. Professor Alexander will present research on XPO1 inhibition in Duchenne Muscular Dystrophy, or DMD.
Thank you, Tina, and thank you, Sharon, for giving me the opportunity to present some of my work. Again, my name is Matt Alexander. I'm an assistant professor in pediatric neurology and genetics at UAB in Children's of Alabama. And simply put, my lab's interested in developing novel therapeutics for Duchenne muscular dystrophy and other rare neuromuscular disorders. Next slide. For those of you not familiar with Duchenne muscular dystrophy, it's an X-linked disorder. It's caused by non-functional mutations in the dystrophin gene. It results in a loss of myofiber membrane integrity. And as a common theme you'll see over and over again, it results also in increased fibrosis and inflammation. Due to the lack of muscle myofiber membrane integrity, there's abnormal calcium regulation, decreased contraction force. Dystrophin itself acts as a molecular shock absorber, and as a result, there's also impaired glucose metabolism and overall impaired muscle function. Next slide. And again, you know, Caesar and Tom did such a great job. I won't beat a dead horse, but you can see a common theme emerging that if you can find ways to block inflammation, block fibrosis, and in the case of DMD, you'll block the disease progression. You'll keep the myofiber membrane intact. You'll keep the animal, or in the end goal, the patient, walking better because the muscle itself is overall more intact. And so one of the things we decided to do first was test out KPT-350, the SINE inhibitor, SINE compound, excuse me, in zebrafish model of DMD. And I won't go into all that data, but basically we identified an optimal dose and were able to show that the KPT-350 was able to correct locomotor function and overall viability of DMD zebrafish. And so based on those initial studies, we decided to expand into another model, the D2-MDX or DMD mouse model, by giving it KPT-350 via oral food pellet. Next slide. And so we came up with a dosing regimen of giving these oral food pellets, these peanut butter balls, 3 times a week for 8 weeks at 5 mg per kg body weight. And we put these mice through a battery of tests. It's known as the TREAT NMD or Treat Neuromuscular Disease Guidelines. And so you can notice that the DMD mice overall have decreased myofiber size, as well as this pattern of degeneration and regeneration of myofiber nuclei. And so one of the hallmarks of degeneration, regeneration is centrally located myonuclei, and that's shown in panel B. And so one of the things we did, again, through all these battery of tests is look at centralized myonuclei. And we observed in the mice, the DMD mice treated with KPT-350, we saw decreased levels of centralized myonuclei, overall increased myofiber size, and overall decreased areas of myofiber degeneration and fibrosis, as shown in the graphs below. Next slide. And so again, one of the things we wanted to look at was different biomarkers for inflammation and fibrosis. And we partnered with another lab, Dr. Armando Villalta at UC Irvine. And so we looked at different populations. We know that in DMD, there's a lot of emerging evidence that there's a pro-regenerative macrophage that competes with anti-regenerative macrophage population. And so you're seeing on panel A, T-SNE plots showing the different populations. And what we were able to observe was that when you gave the mice with DMD, the KPT-350, they overall shifted from an anti-regenerative macrophage population into a pro-regenerative M1-like macrophage population. And we saw also decreases in overall inflammatory cytokines as well, and things like, you know, things that Tom mentioned, IL-6, TNF-alpha. And so these are drivers of inflammation. These are causing the myofiber membrane to break down in DMD. And so this was very exciting to us. And so overall, next slide. If we can generate broad conclusions, we were able to observe that if you give mice or zebrafish KPT-350 or SINE inhibitors, you can block myofiber membrane degeneration. And so again, through different histological markers and biomarkers, we were able to show that if you give these DMD mice KPT-350 or a SINE inhibitor, we were able to overall improve locomotive and muscle function. We were able to improve muscle force and decrease inflammation, as well as promote this anti-inflammatory or pro-regenerative macrophage population in our DMD mouse model. And so this work was recently published in Molecular Therapy in Hightower et al. In 2020. And so with that, I'd like to then pass it back to Tina.
Thank you, Professor Alexander. Finally, Dr. Clifton Moe of Dana-Farber Cancer Institute will be speaking on the role of XPO1 in cancer.
Thank you, Tina, and thanks everybody for listening. I'd just like to briefly touch upon the XPO1 story in oncology, which has already been essentially a successful bench-to-bedside story with plenty of potential remaining for further advances. So near and dear to my heart within the multiple myeloma space is where a lot of these seminal nuclear export work was done preclinically. And on the left here, we have something that looks very similar to what you saw in Cesario's discussion. But this is a study on the MM1S multiple myeloma cell line after exposure to KPT-185. And as you can see, the P53 and ICAP-B tumor suppressor proteins demonstrated significant nuclear localization very quickly within as early as 2 hours after exposure to KPT-185 with more downstream cell regulatory proteins such as P27 and FOXO3A demonstrating nuclear accumulation within hours afterwards. On the right, we have another representation of the mechanism of action of XPO1 inhibition in the cancer cell, which involves, importantly, both the restoration of functionality of multiple tumor suppressor proteins to include, but definitely not limited to, P53, as well as, importantly, the reduction in translation of multiple oncogenic mRNAs, which would ordinarily be translated in the cytoplasm and are now trapped in the nucleus to include c-MYC, BCL2, survivin, and others. Next slide, please. So just another schematic of the function of XPO1 in terms of shuttling proteins between the nucleus and cytoplasm. Again, just to reiterate what was presented in the previous presentations, in normal cells, XPO1 is known to maintain homeostatic levels of these proteins and potentially oncogenic mRNAs. However, in the cancer cell across multiple cancer subtypes, as I'll show in just a second, the action ceases to become homeostatic and becomes, frankly, oncogenic. XPO1 plays a key role in multiple cancer-related processes to include cell cycle progression, apoptosis, as well as metastasis of primary solid tumors and drug resistance across multiple cancer subtypes. It is important to note that overexpression of XPO1 has been demonstrated across multiple types of cancer, both within my realm in hematologic malignancy as well as in multiple solid tumors. Next slide, please. So again, within the myeloma space, in a murine model, there was definitely a lot of promise demonstrated by exposure to SINE compounds. On the left, both in terms of mouse survival, and importantly on the right, in terms of decreasing bone damage. So at least within the multiple myeloma space, in addition to the cell regulatory and pro-apoptotic effects of SINE compounds, there also seems to be a direct reduction of osteolytic bone damage, most likely due to inhibition of osteoclastogenesis. Next slide, please. Importantly, within multiple cancer subtypes, it's becoming evident that although the XPO1 inhibition is very toxic to the malignant cell, it does not appear toxic to normal hematopoietic cells in the animal models. The example demonstrated here is in the FLT3 internal tandem duplication AML leukemia-graft mouse model, where you can see exposure to KPT-251 results in a logarithmic reduction in mean bioluminescence, as well as a significant increase in mouse survival. And on the right, you have a histologic representation of preservation of normal hematopoietic cells after exposure to KPT-251. So again, this appears to be much more toxic to cancer cells, as we definitely want it to be, as opposed to more toxic to normal important hematopoietic cells. Next slide, please. And now we are towards the right-hand side of the bench-to-bedside story. This is from a seminal phase 1 study of XPO1 inhibitor therapy in multiple myeloma. And as you can see, pre-treatment on top compared to post-treatment on bottom, you see significant increase in nuclear localization and expression of the important p53 gene. To the right of that, you see a definite increase in the number of apoptotic cells as measured by the apoptag stain, as well as multiple other important tumor suppressor protein levels that are expressed increasingly after exposure to XPO1 inhibition. Next slide. Also very promising in terms of XPO1 inhibition is its increasingly demonstrated ability to partner well with existing anti-cancer drugs within the realm of myeloma. It appears that the XPO1 inhibition is the potency of it is enhances the potency of the proteasome inhibitor, which is one of our staple drugs for multiple myeloma that has definitely helped to transform the treatment landscape for this disease. This is thought to be mediated primarily by an enhancement of NF-kappa B inhibition, likely due to the effects on the I-kappa B nuclear localization, as demonstrated in the previous slide. Next slide, please. So despite my urge to talk for the next hour about multiple myeloma, I should mention that it is not just myeloma that XPO1 inhibition is relevant. Across multiple blood cancers as well as solid tumors, XPO1 overexpression has been demonstrated and importantly has been associated with poorer survival. So again, compared to normal non-cancerous cells, cancer cells have greater XPO1 expression, but even within a cancer itself, a higher XPO1 expression seems to be a definite negative prognostic indicator and deleterious to the patient compared to low XPO1 expression. Next slide, please. Fortunately, in multiple preclinical models, SINE compounds have demonstrated robust anti-cancer activity across all of these listed blood cancers and solid tumors. You know, in the oncology space, where sometimes there appears to be somewhat of a disconnect between hematologic malignancy and solid tumor oncology, this appears to be one target that is shared across multiple tumor types, both solid and liquid. Next slide, please. And that's a hopefully less than 10-minute run through XPO1 inhibition in cancer to date. Thank you so much.
That was fantastic. Thank you so much, Dr. Mo. And thank you to all of the panelists for your insights on the role of XPO1 as the mechanism in a variety of disease states. This was a very, very informative afternoon together. So with that, I'm going to turn it back over to Dr. Shacham.
So thank you again for all the panelists and for all our guests today for staying for our 60-minute webinar that actually will be just around 75-minute webinar, something for us to learn for our next Karyo Talk. And I want to just summarize again with our cartoon looking at the XPO1 train that has many cargoes outside of the nucleus to the cytoplasm, which results in the loss of key important superheroes that's supposed to be in the nucleus, and keep an eye on the DNA and make sure that the cell is either not grow as a cancer cell or not fall apart in a neurodegenerative disease. And to prevent this out of control train, we have the SINE compounds here that can stop the train and let the guardians of the genome do their job. So I hope we were able to explain a little bit about the communication between and the transport between the nucleus and the cytoplasm, and about the role of the nuclear pore complex and the XPO1 in the different disease. So thank you very much, everybody, and have a good afternoon.
Thank you. And one parting note, we are excited to share that there will be future webinars. So please be sure to follow us on social media and to check back on our website to find out about future events like this one. Thank you.